So to kick off the program, I'd like to welcome up Dr. Charles Sawyers, AACR President, to welcome us all and begin the program.
Thanks, Andy. I'm a faculty member at Sloan Kettering. I'm the president of AACR, and I want to thank Andy and AdvaMedDx for partnering with us on this conference. This is a very timely topic. As you know, the investment that the NIH and NCI and others have made in cancer research has led to an explosion of molecular information. We're going to hear a lot about the application of that in the early stages this morning. And I think one of the pressing issues we're going to deal with today is can we catalyze the speed at which this information gets translated into commercialized diagnostics that can be used by the world of cancer patients and physicians. So without further ado, I want to welcome our keynote speaker. I'm very proud and pleased to have my friend and colleague Harold Varmus, the director of the National Cancer Institute, kick off the meeting with some thoughts about this general topic. As I think Harold needs no real formal introduction prior to his taking on the role as director of the National Cancer Institute. He was my boss at Sloan Kettering. And Harold, welcome. Thank you.
Thank you. Well, thanks, Charles. So I appreciate a chance to come and talk a little bit about diagnostics in general. As Charles has indicated, this is a hot topic now in our new approach to cancer, but I think it's worthwhile spending a couple of minutes at the outset talking about diagnostics in general. I'm sorry, I'm not apologizing, but I'm sorry that there's only one small slide and the thousands of dollars that went into this meeting. We couldn't have another projection over here and something that would allow people in that corner to see, but so be it. So I want to say a few things about the centrality of diagnostics and the meaning of a diagnosis, and then talk a little bit about some of the new opportunities that I think many of you are aware of for improving diagnostics, especially in the cancer arena, and then survey fairly briefly, because I only have 20 minutes or so, the requirements for and the obstacles to achieving the improvements that we all can foresee in diagnosis of cancer. So let me talk first for a moment about the meaning of diagnosis. And this really is a much more complex category of the things that we do in medicine than might first meet the eye. In some ways, diagnostics are central to deciding what to do with a patient, and they also reflect our underlying understanding of what goes wrong with our bodies. So first, in the sort of the most obvious respect, diagnostics tell us what we can do to control disease, and I want to use that word control fairly broadly. So in the world of cancer, of course, diagnosis traditionally begins with identifying where the abnormal cells are in the body, done by various physical exam and by imaging and other things, and then by looking histologically at the nature of the lesion and try to make some predictions. But what we're here to talk about today are the ways in which our deeper understanding of cancer, achieved through genetic and molecular studies, changes the way we approach diagnosis and the way we understand disease. And that can be done by using things we call biomarkers, by looking at genetic lesions with next-gen sequencing and other molecular technologies, by looking at circulating cells and circulating DNA, and all these things become part of the diagnosis. So there's a pragmatic aspect to diagnosis that's very important. What is the disease we're dealing with? That is when a patient has symptoms, what is causing them. But it's, I think, legitimate to say, in the case of someone who doesn't have symptoms, is there a risk of disease? And again, the same kinds of molecular technologies can be applied. Think BRCA1 mutation testing to allow us to say whether a healthy person should now have additional tests, screening, and possibly even surgical procedures to control the risk of cancer. And these diagnostic categories inform what we tell a patient with respect to the severity of the disease, the likely outcome, and most importantly, they tell us what we should be doing to treat the disease or to prevent the occurrence of disease in someone who is diagnosed to be at high risk. But I'd like to extend this notion a little more broadly to a sense of philosophical, a knowledge content perspective, because having diagnostic categories reflects our understanding of disease. It illuminates what we know about the causes of disease, the mechanism by which disease arises, and to a certain extent, what we have been able to achieve with respect to opportunities to prevent and treat disease. These ideas have been encapsulated in a report that Charles and others wrote for the Institute of Medicine and the National Research Council a few years ago called Towards Precision Medicine. And with this diagram, which is, I hope, now fairly familiar to most of you, what this tells us is that the knowledge that we generate in the lower left corner, for those of you who can see these slides, generates new notions about disease that changes our conception of disease and creates what we call the taxonomy of disease. That is, what is a disease, what separates one disease entity from another, and what is useful to know about that disease in order to make steps to control it. This taxonomy allows us then to say whether a certain patient fits within a taxonomic category. It allows us to make a diagnosis that's more accurate than what's been happening traditionally in cancer care. It then allows us to take on a treatment regimen that may be more appropriately targeted to that specific category of disease. Hopefully that leads to greater outcomes. And these products of clinical investigation, if properly aggregated and stored and analyzed, can lead to a growth in knowledge that then drives further refinement of these taxonomic categories. So this goes to the heart of what medicine's about. It's the way in which science connects most profoundly with patient care. The understanding how this system works and how it can work better is what connects the world of science to the pragmatic objectives that were recently outlined for you at the start of this meeting. So, what I'm going to be trying to convince you of in the next 10 or 15 minutes or so is these simple components of an underlying thesis. First, that knowledge box, our knowledge about cancer, is growing faster than our validated, accepted, reimbursed diagnostic tests, that all of the steps that are in the developmental pathway for improving diagnostic categories and diagnostic tools, that is basic and applied research, design of tests, the testing of tests, the implementation of those new tests, all must be supported across a broad spectrum of activity to achieve the potential that we believe that cancer research now offers for patient care. Nevertheless, despite these hopefully inspiring words, there are a lot of problems. The developmental pathway for diagnostics is complex, and many components of it are not well supported as yet. Why is that? Well, I think there's some underlying reasons that may not be self-evident, but we don't place a lot of value on these tests in general. Most physicians don't think through what a diagnosis actually means, and the validation, the valuation that we place on development of such tests may affect the way we reward efforts to promote the development both in the academic and even the for-profit sectors. So in the academic sector, if you say to your colleague, I'm trying to develop a diagnostic test, that may be viewed as too applied and not worthy of promotion and exaltation. In the for-profit sector, it's recognized that compensation for these tests is not great. And there's a simple statement one can make about this, that a simple diagnostic test that might cost just a few dollars could keep someone from going on a regimen with a targeted therapeutic, much more highly valued, that might cost $100,000 a year, and also prevent that patient from going on a more appropriate and cheaper drug. So there are valuations here that influence the likelihood of making some progress. So given all that, I'm going to say a fair amount about the kinds of things that we need to support in the scientific community and the clinical research community to make make better progress. And I'm going to review about 6 of these categories, somewhat arbitrarily selected to emphasize my own predispositions. So the first thing I think is important to do is to recognize the significance of diagnostics. I've tried to give you some theoretical reasons for doing that, but let me be a little more pragmatic. So there are at least 2 kinds of significance that are important. The first is the medical, the other is the economic. And by that I mean not just the need to reimburse these things more vigorously, but also to recognize the economic implications of having an appropriate diagnosis as a cost saver. So my own lab happens to work on lung adenocarcinoma, so I'm going to use an example from that. But I think most of you in the room know that over the last decade or so, we've had a transformation in understanding of lung adenocarcinoma, a disease that some people still refer to by the totally vaporous name of non-small cell lung cancer, totally ridiculous diagnosis. But we also now know that it's important to say not just lung adenocarcinoma, but we need to say what driver mutations that may be targetable mutations are present in those tumors. And as long ago as 10 years ago, the only mutation we knew was RAS, which is not directly targetable, but it influences one's choice of therapeutic. But since 2004, we've had mutations in the epidermal growth factor receptor, which allows effective, if transiently effective, therapy with tyrosine kinase inhibitors. And since then, there's been, with the rise of more effective sequencing methodologies, the identification of lots of other mutations, some of which are targetable with specific drugs like ALK translocations and BRAF mutations, but others that are potentially targetable have not yet achieved a drug approval. But you can see that we've rapidly transformed the landscape in this disease over the last several years. Now, this has strong medical implications for those who have mutations that can be targeted, but there are also economic implications, and those are illustrated by a study done by the French Cancer Institute, INCA, which has, in France, everyone with lung cancer gets tested for certain mutations, including epidermal growth factor receptor mutations. And INCA showed a few years ago that by testing essentially everybody in France with a newly diagnosed lung adenocarcinoma, one saves, while spending 1.7 million for the tests and 35 million euros for gefitinib, one of those tyrosine kinase inhibitors, the same time for treatment of patients who have epidermal growth factor receptor mutations. You also save 69 million euros by not treating people with gefitinib to see if they respond. So this cost effectiveness is the kind of message that is not easy to ascertain, but once it is, should be made apparent to people who are responsible for setting reimbursement rates and overseeing the implementation of diagnostic methods. Second issue has to do with a sort of obvious issue, has to do with the need to support a variety of basic oncological studies and preclinical science that informs our use of diagnostics by simply deepening our understanding of cancer. Now, I showed you a very simple diagram of the important driver mutations that we now recognize as emblematic of certain types of lung adenocarcinomas. This is an intermediate stage of understanding lung adenocarcinoma, showing that there are a lot of other mutations in genes we don't understand very well, various combinations of genes, a level of heterogeneity in these cancer cell populations that we don't understand well. And what this shows, I neglected to say, is a series of lung cancers across the horizontal axis and genes that are mutated recurrently among a fairly small number, a couple of hundred of lung adenocarcinomas. And to understand this kind of data and to make use of it clinically, there are a lot of things that need to be done. Bigger cohorts to identify still other genes that aren't on that list, attempt to understand the functional consequence of the mutations, which in some cases may be very limited, in other cases profound. An effort to understand what combinations of mutations mean and whether mutations mean different things in the context of the germline genome. And then the additional sort of obvious things, how do you inhibit cancers that are influenced by certain types of genes. The target may not be the mutant gene, it may be something else. And then how do you use that information to predict the course of disease, to determine whether or not there's going to be a therapeutic response, whether there is a likelihood of drug resistance, and so forth. So there's a lot of additional work to do. Sequencing is not the end of the story, and our Center for Cancer Genomics at NCI is wrestling with those issues. But the kind of research that needs to be done extends well beyond the most obvious stuff, genomics and signaling pathways and so forth, to the notion of what kind of diagnosis are we actually making. One of the biggest problems we're facing in cancer care today is overdiagnosis, finding an elevated PSA, for example, and intraepithelial neoplasm, and knowing whether or not that should be treated vigorously, not at all, or an intermediate way. And these kinds of early lesions that may or may not be stepping stones to invasive and lethal cancers represent a diagnostic dilemma that I hope we'll be able to solve. It's one of our provocative questions in that PQ initiative we've been sponsoring. This will also be the topic of a request for applications that we're going to be issuing soon to try to understand better ways to make a more precise diagnosis of, that includes some indication of how vigorous treatment should be in pancreatic, breast, prostatic, skin cancers, and others. Being able to carry out tests of diagnostic procedures and learning which drugs are going to work in which molecular contexts is obviously very important. And our clinical trial system at the NCI has undergone a lot of changes to try to accommodate our interest in using diagnostics in more powerful ways. So as many of you may know, we have been transforming the so-called cooperative groups that carry out most of our advanced stage trials. Under the leadership of Jim Doroshow at the NCI, we've been emphasizing a number of centralized aspects of the clinical trial system that is in many ways engineered to promote a greater understanding of diagnosis. That includes biobanking of samples from the tumors of all patients enrolled in these trials, having means to get various kinds of omics tests, not just sequencing, but other things done on those tumors when necessary. We are urging the cooperative groups to incorporate molecular tests to refine diagnosis in protocols. We have a series of trials that I don't have time to describe in detail. I'll show you one that rely heavily on using molecular tools to categorize tumors before therapy is chosen. And then we placed a lot of emphasis on retrospective analysis, and you're going to hear from David Sollett in a couple of moments about his beautiful work with exceptional responders, and we're trying to amplify that kind of approach on a much bigger scale. This is a picture of the so-called match trial, and I don't want to go through this in detail, but I'll point out that the first step in this trial is not to say what organ is affected, but instead to carry out genomic sequencing on tumors that have failed to respond to rounds of conventional therapy, to try to find mutations that dictate a certain action. We've gathered up drugs, most of which are not, in fact, essentially none of which are approved for use in these indications, to test them in patients and to determine whether or not patients have progressive disease, stable disease, or a remission, and to validate remissions by taking folks off drugs and so forth. And this kind of protocol is a revolutionary step in the way we approach clinical trials, and hopefully will allow for a much more complex and sophisticated approach to diagnosis and appropriate therapy. All of you who follow the conduct of research, especially biomedical research, whether you're reading Nature magazine or The Economist, are aware that there are problems in the development of information that applies to the use of these new classes of tests, molecular and genetic and so forth. And There have been now many reports from the Institute of Medicine, the most important of which from certain points of view is this more recent one that the NCI requested a couple of years ago on the use of various kinds of omics testing, whether it's gene expression arrays or DNA sequencing, in the testing of diagnostic protocols that could be employed in clinical work, but before we subject patients to the risk of having their disease determined by these methods, we need to feel that they are safe and, more importantly, reliable. One important step in trying to make secure use of these new methods is to take advantage of the checklist methodology. And a paper from Lisa McShane and other of her colleagues at the NCI recently appeared in Nature and in BMC Medicine, references provided here, that lay out 30 criteria that need to be applied with respect to sample accrual, the methods themselves, the interpretation of methods, the statistics, and I urge all of you who are thinking about these kinds of methodologies, most of which do depend on so-called omics technologies, to look at this checklist, because this is a very useful thing to do. And it's not totally new. Indeed, some years ago, Lisa and her colleagues developed the so-called REMARK guidelines that are used for reporting efforts with biomarkers to deduce prognoses. But again, the whole idea is to have a regimented approach that attempts to avoid the pitfalls of these tests, which have a reputation, unfortunately, of being unreliable, and they need to be as reliable as possible before we use them on our patient populations. The fifth category of need has to do with some of the regulatory and approval issues that Charles mentioned briefly, and I'm not going to prescribe on this front, but I want to point out that if we're going to have a confident industry here that feels that the investment they make in new diagnostics will be appropriately treated, we need to have some answers to some of these questions. For example, we need to say who's going to recommend that a test, who's going to speak authoritatively about recommending a new test, and there are a lot of contenders listed here. There's going to have to be an approval process of some kind that tells physicians what is appropriate to use and what has been adequately validated. At least I believe there should be an approval process. I'm not enthusiastic about companion tests because we all know that in due course we're going to be doing these tests in an aggregated way through high throughput methodologies, and assigning a companion test to each drug that we use in advanced therapy is, in my view, not going to be workable. I think it is sending a good message at the moment, but we need to be transitioning to something that is different in style. Oversight to be sure that the tests are properly conducted is important. I don't know if we're doing it in the absolute best way at this point. Certainly, I lived through an experience at Memorial Sloan Kettering that was very difficult. We were using mass spec to look for EGF receptor mutations, and every time we wanted to look for a different mutation, we had to go through a state process that involved testing 50 positives and 50 negatives and show we could identify them all, even though the methodology was exactly the same each time. So these are cumbersome and inhibitory to adapting tests to new understanding of disease, and that's becomes a barrier to implementation of tests. Importantly, who's going to be doing the reimbursement? That's often the real gateway here. Right now, reimbursement practices seem to be pretty loose, but as we worry about the expenses of healthcare, those gateways could be more constricted. And there are obviously a lot of players, and having some kind of uniformity in reimbursement practices would be great, although I'm not sure it's possible in our system of healthcare. And then important to me, as someone who supports the National Cancer Institute and would like to see the products of our labors used widely, who's going to be using these tests? And we all know that uptake, for example, of EGF receptor mutation testing has been relatively slow. It tends to begin in research, with research labs that do the work, and gets used more and more widely among the NCI-designated cancer centers. But those cancer centers treat only 15% of cancer patients in the country, and it's very important that networks of oncological practices, community hospitals, and cancer centers also use these methods when they become demonstrated to be important in patient care. Finally, I think it's important that we look at stories about the situations in which diagnostics could play an important role, and look for reasons why efforts to build diagnostic tests that are widely used have been successful or have failed. And I have asked my colleague Lou Stout, who's the leader of the Center for Cancer Genomics and a well-known investigator in the world of lymphomas, to provide me with one concluding example of how diagnostics are being or not being taken up in the medical community. So Lou has been working for a long time on a class of tumors that has classically been defined as diffuse large B-cell lymphomas. They represent a large fraction of the non-Hodgkin's lymphomas. There are over 20,000 new cases a year with about a 50% cure rate in the US. This whole class of tumors histologically defined by the kind of pattern you see in this one histological picture. But he showed now 13 years ago that a simple gene expression profile can distinguish at least two well-characterized types of tumors, one that's called ABC for activated B-cell-like, and the other germinal center B-cell-like, or GCB. And these expression arrays, now probably viewed by most of you multiple times, are highly persuasive. And what's more important, because a pretty picture doesn't mean much to a patient, these patterns are predictive of the kinds of mutations one's likely to find in those tumors. And here are lists of some of the genes. So some of these are, as we say, actionable. They are predictive of whether or not patients are going to respond to conventional chemotherapy, with the outcome in ABC type much less good than for the GCB type. And they are predictive about responses to certain targeted therapies like ibrutinib, which is a tyrosine kinase inhibitor. And you can see that over 40% of patients with ABC type disease respond to ibrutinib, and very few patients with the GCB type. Nevertheless, we still don't have a standard diagnostic test for these subtypes. The subtypes are recognized. There are some organizations that endorse the use of a poor man's histochemical test that is arguably a lot less good than doing some kind of array procedure for gene expression profiling. But nevertheless, after 13 years, we still have a disappointing outcome with respect to the diagnosis of this relatively common cancer. So I remind you of what I've tried to remind you of, namely the kinds of things that are needed to make diagnostics as powerful as the underlying science of cancer that we've achieved over the last couple of decades. And I hope that during the course of this meeting that some of the more thorny questions about how we get higher valuation on the kind of things that are needed to advance diagnostics will be achieved. And I look forward to hearing from many of my colleagues about what you're able to discern during the course of the meeting. Thanks very much for your attention. I don't think anything I've said is highly questionable, but if you have any questions that are not about the NIH budget, I'd be happy to hear them.